This is part 1 of embryo culture 1.0. Then the embryo culture 2.0 will be taken by Dr. Lakshmi. So I will be covering the basics of culture systems today. Now coming to the first slide, the goal of embryo culture is to replicate the conditions of maternal reproductive tract. So we have to replicate the conditions of uterus, the fallopian tubes, and that is how we are able to achieve, optimal growth rate in the embryos. So if you look at the journey of an embryo, you have to understand the journey of a person or anything, in order to replicate it. So if you look at the journey of embryo, it's a unbound suspended organism. It's a very small group of cells, and the pre-implantation development it ranges from fertilized oocyte to hatched blastocyst stage over a period of 5 to 7 days. And its nutrition is purely dependent on cytoplasmic stores of maternal origin. So initially, when the embryo grows, it is purely dependent for its nutrition on cytoplasmic stores of maternal origin. And then when it reaches the uterus and the oviduct, it derives the nutrition from the oviduct. So composition of these secretions differs as the fertilized oocyte progresses through the fallopian tube and ultimately it moves to the uterus. So when we try to design a culture system, we have to use the same composition of culture media as it is in the uterus or as it is in the fallopian tube. And it's very difficult because these are very tiny structures. So it is very difficult to, uh, sacrifice the uterus or sacrifice you can't do it on humans. So you have to, and small animals have got even smaller organs. It is very difficult to decipher the composition of these secretions. First of all, to get these secretions and then to find out their composition. It's a very difficult task, but scientists with a lot of devotion and dedication they've done it. So if you look at the embryonic development, the reproductive tract secretions influence the various events in the embryonic development. Number one is the fertilization. Number one is the maturation, fertilization of the oocyte, then the formation of gamete, which includes, the entry of sperm and then subsequently zygote formation. Zygote is uh, you can say a 2PN structure. When the oocyte is fertilized, it is known as zygote. Then comes the stages of cleavage. It starts on two-cell stage and it goes up to the morula stage. Then comes the three stages of compaction, decompaction, and recompaction. And during recompaction, there is a differential cell allocation. In a morula, if you look at it carefully, some cells will get aligned to the outer cell mass, whereas some cells will form the inner cell mass. And this is facilitated by the formation of a cavity which segregates the inner cell mass and outer cell mass. In between, there is a cavity. And this cavity expands. Ultimately, it becomes an expanded blastocyst. From an early blastocyst with a small cavity, it becomes an expanded blastocyst, ultimately pressurizing the zona to give way. And finally, you see a small herniation of the blastocyst. And after herniation is there, the whole the embryo proper comes out of the small herniation which is there. And ultimately, when it is out of the zona pellucida, it is ready to implant now. So, as I told you, the culture media was brought up or discovered with a lot of great deal of efforts and dedication. If you look at the first media was designed by Ringer, the Ringer's lactate media. It was based on blood culture serum, and it was able to support the development of a beating frog heart. So when we used to do experiments in our school days and college days, we used to use Ringer's lactate to support a beating frog heart. And it was way back in 1882, Ringer gave the formulation. Now, from 1882 to a present situation when the culture media contains up to 80 components, including nutrients, vitamins, and growth factors. And Dr. Lakshmi will be focusing on the micro components and the additives which have been added now, the hyaluron growth factors, insulin-like growth factors. So many things she will be discussing in the next session. But before that, if you look at the key developmental points in the evolution of culture media, Viton is known as the father of embryo culture media. It can support mouse embryos up to blastocyst stage. And then came the discovery of Strepto in 1971. He could culture human embryos up to blastocyst stage using ham-Sefton media. This was a landmark event. Strepto, who developed the ham-Sefton media. But the first baby was born in Earl's simple salt solution, or as we call it, ESSS. This was pyruvate supplemented with patient serum. And step two, and Edwards cultured the embryo in this Earl's simple salt solution, which gave birth to Louis Brown, the first IVF baby. Then came a landmark development in which Gardner he said, no, we have to have two medias, one which is simulating the development in the fallopian tubes, and the other which is simulating the development in the uterus. So he gave the medias as G1 and G2. That is a sequential media. So ultimately, now we are again back to single step media. So there is a concept of single step media and sequential media, which we will just touch upon. Harper, who is a very great scientist from England, she in 2012 she said that it remains true that there is no culture media available that is truly optimized for human embryo development. So it was agreed, and it was admitted that there is we have not been able to develop a culture media which is truly optimized for human embryonic development, because most of the research has been on animal embryos, especially mouse embryos. So we are just trying to extrapolate the developments in a mouse embryo to human embryo. So we are still not there. Now I would like to introduce the concept of a broader culture system, and we'll move ahead of culture media. The culture media alone cannot sustain, like Surya said in the beginning, that it's about culture media, but it's not about culture media. It's about culture system as a whole, and culture system includes culture media, which is a important but a tiny component of culture system as a whole. So when you talk about culture system, the culture system is much more than media. It is about plasticware, it is about oil, oxygen, CO2, gases, light, and we have to minimize insult to the and stress to the gametes and embryos and maximize their embryonic development and the developmental potential, as well as which will facilitate implantation potential. So the role of culture system is to maintain viability of gametes, number one. Sperm function, capacitation, acrosome reaction, everything has to take place almost like it happens in the natural circumstances. It should facilitate oocyte maturation, and it should preserve oocyte competence. After fertilization, it should be decreasing the embryonic stress by taking control of temperature, pH, and CO2, which is looking beyond the media. It should ensure embryonic development and increase implantation rates. This is the whole ecosystem, which is known as culture system or embryonic culture system. If you look at this figure, which I've picked from Gardner's textbook, he has very beautifully elaborated that there are patient factors, there are stimulation factors, genetics, diet, endometrial receptivity, luteal phase support. But in between, almost 80% of the variables belong to the lab. How well trained the embryologist is, how many embryologists you have, how many incubators you have, what is the air quality in the lab. If you're talking about culture system, how good the plastics are, what is the gas supply good, is the oil overlay being done, which culture media you're using, how are you culturing. So many variables exist in the culture system. So just to enumerate, it's the media, macromolecules, gas phase, type of medium overlay, culture vessel, that is the plastics, incubation chamber, that is the incubator. Ambient air quality is also very important, because if you have to spend on something in lab, number one should be the air quality. Anything else will be secondary to it, if you ask me. And even the embryologists themselves, they are part of the culture system as a whole. So the concept of embryo culture system, it highlights the interaction that exists not only between the embryo and its surroundings, but also between all the parameters in the IVF lab. But these are very important words which I'm saying. So you have to understand that everything in lab interacts with each other, and you have to control each and everything in the lab. So just a brief, this slide says that total factors in the lab which are to be controlled are 175, and half of these factors you control even without knowing them. The chief factors are, like, just few examples are air quality, pipettes, hyaluronidase, plastics, denudation, temperature of surfaces, light, culture medium, dishes, protein, supplement. So many, as you read more, you come to know more. And then the processes are to be controlled: retrieval, sperm collection, oocyte preparation, sperm preparation, conventional IVF, ICSI. n number of variables are there in an IVF lab. So it doesn't look, it is not that easy as it looks. And this beautiful article by Cairo Consensus Group, it wrote in just one line, it said everything. It said that there is only one thing that is truly important in the IVF lab, everything. So in short, they said that you cannot ignore anything in the lab. You have to be on your toes all the time, and everything is important in the lab. And so is the clinics actually, but then clinics, we are still not there, I think. In IVF lab, there is a huge quality control. There are so many variables which have to be controlled. But in clinics, there are variables, but there are no defined KPIs, or there are no defined PIs also for an IVF clinical practice. And I think we have to develop them. Because many a times there are variables, there are uncontrolled things in the clinics, and it reflects in your laboratory practice. So when I talk about culture system, I always like to say that it is the culture media, consumables, culture conditions, number two. Number three, the protocols, how the protocols you're adopting in the lab. Embryonic assessment is very important. And the overall quality control is extremely, extremely important. When I talk about culture media and consumables, culture media, as Dr. Surya also said, it's an artificial environment for the embryo. It mimics complex environment of uterus and fallopian tubes. And we discussed in the previous slide also, it should be able to support sperm function, oocyte-mediated cleavage, fertilization, genomic activation, and early stages of embryonic development. When I look, since Lakshmi will be focusing on, so I'll be just running through culture media in general, because Aanantha Lakshmi is a renowned teacher. She will be taking up these subjects. So if you look at the broad composition of culture media, it is number one, salts, energy substrates, pyruvate, protein. There's a complex mixture of amino acids and glutamine in the form of dipeptide. I'll come to this later. Phenol red, optional. It may be there, it may not be there. You need to add antibiotics such as penicillin, gentamicin to prevent infections. And there has to be a buffer to prevent against the pH changes. pH might change due to temperature, it might change due to CO2 concentration. So these are important. This is the backbone of a culture media. Salts, energy substrates, protein, amino acids, glutamine, then antibiotics and buffer. Very simple it looks, but it is very complex. When you look at the energy substrates, you have the options are pyruvate, lactate, and glucose. Pre-compaction will prefer pyruvate, whereas post-compaction will prefer glucose as the energy substrate. So most of the medias have got EDTA, which prevents the utilization of glucose during pre-compaction stage. But in the post-compaction media, EDTA is omitted. So post-compaction embryos will preferably take glucose as the energy substrate. The energy substrates, we'll discuss about the amino acids. They are buffers of pH. They are antioxidants, chelators, and they have signaling mechanisms. And the inclusion of amino acids in the culture media enhances embryonic development to the blastocyst stage. So there was an experiment carried on when amino acids were included and they were not included. It was found that with the right amino acid at the right stage, the blastocyst development was much better. There are two kinds of amino acids: cleavage amino acids, which is required for pre-compaction embryos, and compaction amino acids, which are required after post-compaction. So the cleavage amino acids have got a zwitteronic nature, and there are strong intracellular buffers of pH, whereas the compaction amino acids have got non-essential amino acids primarily, and also glutamine is there. So the addition of glutamine to simple culture media significantly increased development of human blastocyst in culture and increased the pregnancy rates. So when the glutamine was added to a culture media, it increased pregnancy rates and the embryonic development also.
Sir, excuse me, sir.
Yeah,
No, full.
You can't see?
No, sir. We can see, sir, but make it full screen or slideshow.
Okay, there's some issue, I think. It's full screen from my side. Okay, I'll share it again. Can you see now, Dr. Lakshmi?
It is visible, sir, but full screen. Yes, sir. Now it is okay, sir.
Okay. Okay, amino acids are buffers of intracellular pH. They have got antioxidant properties. They help in signaling mechanisms, and their addition increases the embryonic development to the blastocyst stage. There are two classes of amino acids: the pre-compaction amino acids or cleavage amino acids, which have got mainly buffering function, and the post-compaction amino acids or the compaction amino acids. And they include both non-essential and essential amino acids. Plus, the addition of glutamine enhances blastocyst formation and increases the pregnancy rates. So, the addition of glutamine to a simple culture media significantly increased development of human blastocyst in culture and increased the pregnancy rates. The essential amino acids, they stimulate cleavage rates as well as they increase the development of ICM on inner cell mass in the blastocyst. A very important component is glutamine and especially L-glutamine, the levo form of glutamine. It it's an amino acid which breaks down spontaneously with ammonium as a byproduct. So when ammonium accumulates in the culture media, it's a big concern, and the effect is magnified in incubators which are supporting 20% oxygen or no, atmospheric oxygen, and incubators which are hypoxic benchtop incubators or hypoxic incubators, they are able to minimize the effect of ammonia concentrations. So ammonia, it alters embryonic differentiation, it alters metabolism, it alters gene expression in vitro, and consistently and significantly reduces the implantation and fetal development rates. So what was designed in place of L-glutamine? A simple solution was the introduction of glutamine in a stable dipeptide form, that is alanine glutamine or glycine glutamine. It does not break down at 37 degree. So when we replaced L-glutamine with glutamyl in a stable dipeptide form, things got solved. Ammonium buildup was much lower, and all the bad effects of ammonia, such as poor bad effect on gene expression, bad effect on metabolism, and decreased implantation rates, were not seen. If you talk about human serum albumin, then we have got albumin, which is the most abundant protein in the fallopian tube as well as culture media. It has got antioxidant properties. It's a scavenger and carrier of growth factors. Unfortunately, when we are trying to develop human serum albumin, there are a lot of batch to batch variations, lot to lot variations, which doesn't provide consistency to culture media. Recombinant HSA or human serum albumin is also available, but it is very expensive. It offers consistency and more tolerance to cryopreservation is provided to the embryos. So, recombinant HSA, you should always look for this presence of this in the culture media composition. Chelators, EDT, I've talked about it. It limits the availability of magnesium as a cofactor for glucose metabolism. So if EDT is present in the pre-compaction stages, it limits the utilization of glucose because in pre-compaction, pyruvate is the main energy provider. EDTA also stimulates development prior to compaction. The presence of EDTA for blastocyst development, it compromises the subsequent development and competence of the embryos. And EDTA,
Can you go back to the next slide, sir?
uh, can you mute, please?
Okay, sir.
So the presence of EDTA has also been linked to the increased incidence of monozygotic twinning in the blastocyst. So if you carefully see blastocyst on day 5 or day 6, many a times you find that you are seeing two inner cell mass. Till the time you are sensitized to seeing two inner cell mass, you will never be able to see. But the day you see it once, you will definitely look for it in a blastocyst. So the theory behind is that EDTA may be responsible for the development of monozygotic twinning. So Dr. Lakshmi will be taking these micromolecules after in the next session. So I'm not going to touch upon. I'm just going to tell you that hyaluronate, antioxidants, vitamins, insulin, so many things are being added in the in the culture media nowadays, and they have shown limited evidence, but in some patients it has definitely contributed to the pregnancies. Also, if you look at the effects, then effects of antioxidants, the effect, it works much better in incubators with 20% oxygen compared to incubators with 5% oxygen. So Lakshmi will be taking all these studies. Similarly, we have got growth factors such as GM-CSF. We have got EGF, insulin-like growth factor, platelet activating factor. So many factors are being, tried upon in the latest new generation medias. Coming to something which is the heart and soul of a culture media, that is the buffer system. The idea is to maintain pH between 7.2 to 7.4. And on the meat, and these molecules are selected based on their pKa value. pKa value is the ability to resist pH change. The pKa needs to be matched to the application because many biological processes only function over a narrow range of pHe, that is pH external. So what we have done is we have added 2 to 3 buffers in a culture media. The spontaneous use of different culture buffers allows for adjustment of pKa values to the desired range. So you can adjust the pK value to the desired range in case you're using multiple buffers. It also helps in reducing the individual buffer concentration and potentially the toxicity. So if a particular buffer system was able to provide you the correct pH at a concentration of suppose 100 units, with the addition of another buffer, both of them can provide the same function, the same pH buffering effect at a concentration of maybe 40 units each. So it prevents toxicity. The most common buffers are bicarbonate, which is very important for the fertilization and sperm capacitation. Then we have got the HEPES or HEPES buffer, and we have got the MOPS buffer. Both of them are very critical in functions outside the incubator, whereas bicarbonate is the chief buffer which is utilized for prolonged embryonic development inside the incubator. It is also the chief buffering system in a human body. So if you look at the advantage of bicarbonate is that it is a physiological buffering system in the fluid surrounding mammalian cells. And a major drawback is there is a rapid increase in the pH when the medium is exposed to air, resulting in impaired embryonic development. It can also lead to cell necrosis if there is a prolonged pH change. To every complicated problem in life, there is always a very easy solution. So the solution here is you have to add a thick oil overlay, which will reduce the exchange of gases. Also, you can replace some amount of buffer by HEPES media. As I told you, we can have 2 buffers or 3 buffers. So HEPES is designed to work outside the incubator and bicarbonate inside the incubator. So when bicarbonate buffered media is brought outside the incubator, the pH changes. But if there is a thick oil overlay, or if there is HEPES or MOFs present in the media, the pH will not change that fast. It will change, but it will have some more resistance. Phenol red was present in most of the medias in good old days when we started working 15, 16 years ago. But now it is not a very favored molecule. It was supposed to, phenol red will become pink when the media was equilibrated, but it was a qualitative test. You could never measure the pH correctly with the help of a qualitative test, and especially when you are working in small drops. When you are working in small drops, then the color between red and pink may not stand out. So it's not a very good idea to add phenol red in the media. It has got some estrogenic properties and it is a source of ROS also. So we don't prefer medias with red color. If you look at the supplies, culture media, and reagents, we have to write the receiving date of these supplies, and also we have to look for the changes, the logistical changes. What was the transportation what temperature during transportation and during arrival? What was the opening date of this media, and what date you opened? What is the expiry date? How soon you could consume the media? What are the storage requirements? It should be exposed to light or it should be kept in dark. There is also a certification of analysis which is provided. So there are many minute details which you have to pay attention to. If you talk about the dish, it's a plastic polystyrene dish. It is said that there is nothing physiological about the culture dish. And there is a lack of plastic quality control in the plasticware we are using. It's disposable and the disposable plastics are characteristically CE marked for Europe and FDA marked for USA. Preferably, they should be MEA tested where possible and endotoxin free with validated clinical evidence. It is important to allow the plasticware to off gas before you start using it. And do not place extra plasticware in the incubator. Many times I've seen that people, they just load the incubators with dishes. So when we go to work and there are already, they have kept some 15-20 dishes in the incubator and they ask us to use the same dishes which they have opened. But there is no point in opening these dishes because if you load your incubator, you are loading the incubator with plastics and you might not have off-gassed it sufficiently outside. So it will off gas inside the incubator and it will spoil the culture conditions, which we don't want. So tissue culture plasticware is subject to variations in quality. There have if in a 4-well dish or in a 8-well dish, a 6-well dish, well to well variations have also been seen in the same dish. So you have inspect all the plasticware for damage prior to use. Any damage, any fragment can expose the embryos to toxins because there's a very thin embryo safe coating on the dishes, and if there's a small crack, it will just break away this coating. So if the dish is cracked, especially the where you're placing the embryos, this dish should never be used. It is also important to store plasticware at room temperature away from direct sunlight, as this can affect the properties of plastic and packaging. So when you receive the dishes in a box, open the box and try to keep the sleeves in a room at ambient air for at least 2 weeks. It will allow them to off gas. Mostly the suppliers have off gas the dishes and then they are supplied, but at times you can't rely on them. So if you find that there is a emission of a smell, a distinct smell or VOCs from the dishes you are receiving or the plastics you are receiving, allow it to off gas in a sterile environment. Any detectable scent or odor in the packaging while you receive it should be noted and dealt with accordingly. These are just the examples of plastic dishes which are available in the market. Ensure that the dishes are IVF grade plastic. And when you look at the quality control, then there might be you should look for MEA testing. You should look for endotoxin free, and there is a test known as Limulus amoebocyte lysate assay. Look for these things on the dish, whether it is FEA approved, CE marked. So dishes with inner non-toxic and embryo-friendly coating are available. So you should always talk to the manufacturers, talk to the salespeople about how the dishes are designed and then only start buying it and using it. A very commonly used dish such as Falcon 1006 dish is not CE marked and it should not be used. Whereas most of the people will do a pickup in this dish. If you look at the IVF grade plastics, if you have got Falcon 3655 and Nung 150265 dishes, they are similar size and they're individually wrapped. Try to try to use dishes which are individually wrapped because they are for prolonged embryonic culture because there'll be no cross contamination in such dishes. And there is no point. I've seen people working in the lids of 35 dish or 60 dish as an inexpensive alternative, but please don't use the lids for any culturing any culture if functions. Well, lids are not generally cell culture treated, and there is no coating on the dish on the lids. Lids are simple plastic material. The lack of surface tension will not only result in flatter droplets, but if you start culturing in the lids, so it might not be a cost effective idea. So cell culture plastics are treated in order to change their surface tension and promote better cell adhesion. So always culture on the surface which is meant for culture. Another invisible enemy for all embryologists and clinicians also is the VOCs. In the ART setting, the most common source of VOC is CO2 gas cylinders. Especially, when the CO2 cylinder is a CO2 is a liquid gas. So when the level goes down, when around 20-30 units of CO2 remains in the cylinder, in the big jumbo cylinder, then the CO2 remains at the bottom, whereas aldehydes will go on the top. So in place of CO2, aldehydes will flow in your culture system, and that is where they start harming the embryos. So whenever you instruct your staff to change the CO2 cylinder when only 20 units of gas is still remaining, ask them to change it. Another source of VOCs is plasticware, as I told you. AHU may be a source of the air handling units, refrigerant gases, like cleaning agents, writing instruments, pen. They're all alcohol based. And VOCs, which are almost 100 to 10,000, 1,000 times smaller than the effective pore size, are not trapped by HEPA air filtration. So VOCs, which are very small, most of the VOCs will be taken care by the air handling units, but which are very small. Like, even the COVID virus was very, very small. It could pass through the pores of HEPA filters. So, but there's a catch here that if the activated carbon is supplemented with potassium permanganate and other oxidizing agents, then these molecules will easily be oxidized and broken into CO2 and water. So that is the reason why potassium permanganate coatings are there in the activated carbon filter, and they help in the breaking down of VOCs into carbon, into CO2 and water. Another idea is you can take this 100 dish, which is a big dish in which we generally do the pickups, and we don't use the 1006 dish or 60 dish. Take this 100 dish, and you can put some oil in this and keep it in the incubator, especially the box incubator. And this oil will has a tendency of absorbing the VOCs. So these are crude methods. You can put activated carbon in your lab. You can buy it from Eureka Forbes. You can buy activated carbon granules, and you can keep it in this dish. But all these things have a life of at least 3 to 4 days, not more than that. After that, they will start emitting VOCs. So always, when they are saturated, remove the dishes from the incubator. Another important aspect, as I told you, is quality control and quality assurance, which is very important for proper functioning of the IVF lab. Some of them are mouse embryo assay. I told you about litmus, abivocyte, leukocyte assay for endotoxins. So when you talk about oil overlay, it helps in preventing evaporation of media. It reduces the harmful effects of, it prevents changes in osmolality, it prevents changes in pH, it prevents temperature fluctuations. And not only all this, it traps number of VOCs. We spoke about silent killers. So the oil has a tendency to trap number of VOCs and prevents them from reaching the media beneath. So it is advisable to use maximum amount of oil in a culture dish. When you talk about handling of oil, oil should be handled in dark, and preferably it should be transported in glass bottles and not plastic bottles. It is susceptible to photo-oxidation or peroxidation, which can damage the embryos. And many times, people store oil for long periods in the incubator. It's not a good idea. Don't do that. As I told you, we have explained the roles of oil. It prevents pH, temperature, and osmolality changes. It absorbs VOCs. When it comes to the storage of media, we should store the media in dark, and we should ensure that the cold chain is not broken. And it is always better to incubate the media and oil through overnight incubation. But it is always better to make dishes rather than, put the media in tubes. You can put the media in tubes, but culture dishes you should make a day in advance. You have to understand that it's an ecosystem. Plastic, media, and oil. It's an ecosystem, and there is a certain degree of equilibrium which sets in these dishes after 14 to 16 hours of incubation, and you don't expect that kind of equilibrium coming in half an hour of incubation, even if the media is incubated. When you talk about culture conditions, it has got culture environment and the lab environment. So lab environment is the ambient environment, and culture environment is the droplet culture, pH and the temperature. So to ensure optimal development in the culture media, the ambient environment has to be controlled. Environmental stresses have to be reduced. When you talk about culture environment, it is temperature, which can fluctuate by zero point 0.1 degree. Oxygen, low oxygen is preferred. pH 7.2 to 7.4, CO2 around 6%, osmolality around 290 to 300, humidity, VOCs and particulates. So these are the culture environment variables. When you talk about the macro environment, we talked about the micro environment. Now we talk about the macro environment. A temperature fluctuation of zero point 0.5 degrees plus minus is allowed, permissible. There has to be a high pressure. Air changes have to be at least 12 to 14 air changes per hour to remove all the VOCs and toxins. Humidity control has to be there, otherwise you will start getting fungal infections. VOCs have to be controlled. I've explained you why, and particulates also have to be kept at minimum. A real-time monitoring of all the incubators and the gadgets should be there. So, and there should be alarm system attached to the culture system to notify you and inform you within time. Temperature, as I told you, is very important, and there have been experiments where people have increased the temperature by a few degrees, decreased by a few degrees, but most of the culture medias support embryonic development at 37 degrees. It is said that testicular temperature is 2 to 4 degree lower, so you can prepare the sperm at 34 degree, 33 degree, but it doesn't work like that. Even the female genital tract is at a lower temperature compared to the body, but optimal for embryonic development, culture media is designed to work at 37 degree. So don't play around with the temperature. Even in the dishes are also designed in a way that some dishes touch the surface of the laminar and the incubator directly. They recover temperature much quickly. And in some, there is a gap, there's an air barrier, and these take a while to, achieve the right temperature. Always use the media with oil and lid. It preserves the temperature for a much longer time compared to oil with no lid, no oil with no lid, no oil with lid. So best is to layer it with oil and cover it with lid, even while examining. These are the gadgets to take the temperature, and this is a study, which is a landmark study, in which they showed that minute changes in temperature will affect the mitotic spindle or the meiotic spindle. It will go on vanishing. So try to maintain the temperature. pH again, 7.2 to 7.1 to 7.2 is the internal pH. External pH is 7.2 to 7.4. It is said that the denuded oocytes do not regulate pH. So when you denude them, you have to keep them in a well-maintained pH, or you have to inject them immediately. If you inject them immediately, the injected oocytes have the property to maintain pH. If there is a change in the pH, it will impede the sperm function. It will compromise the embryonic the oocyte competence. It will impair embryonic metabolism. It will affect the gene expression, and the cell number of blastocysts will go down if there is a change in the pH, ultimately affecting the implantation properties of the embryo, and it can also lead to fetal growth retardation. So, so this is about pH, and it is also said that when you warm an embryo, it takes a while to regulate pH. Initially, it has no property to maintain the pH. So you have to maintain the pH externally, but the internal pH takes around 3 to 4 hours to recover. So if you look at the pH stabilization graph, it is said that the pH passes through the phase of equilibration, set point, and stabilization, and takes around 11 to 12 hours for the pH to stabilize in a media drop. If it is a drop, it stabilizes faster. If it is a big it's a well of media or around 500 microliter, will take longer time to stabilize compared to a 50 microliter drop. So the length of equilibration time is very important. Minimum 12 to 14 hours have to be given for pH to stabilize. Minimize openings in incubators. use IR sensors compared to TC sensors. The location of HVAC or the air handling unit outlets is very important. There should be no temperature fluctuations. No cold air should be falling on the incubators. The volume of media and oil is important. As I told you, more media, the more the time to equilibrate. Less media, it equilibrates faster. If you are working in Kashmir, you require more higher pressure of CO2 compared to when you're working at sea level. So higher the altitude, more CO2 is required as the air is thinner there. So if you're working in small drops, then regassing takes much uh, regassing takes much the re-equilibration takes 20 times longer than degassing. So when you're working in small drops, the pH changes much faster and the equilibration is also faster. But again, if you're working in big drops or wells, the pH stabilization is much more, and you should not go more than expose the dish more than 2 minutes, it is said, because once pH changes, it takes around 30-40 minutes to recover. So you should not keep the dish outside. Even if you're keeping the dish outside, keep it covered with oil as well as the lid when when you're examining the embryos under a microscope. So 1 degree of change in CO2 will change the pH by around 0.1%. Rule of thumb: increase CO2 1%, decrease pH by 0.1, and vice versa. Osmolality: different medias have different osmolality. Generally, 260 to 290 is the norm, milliosmoles. It is good to maintain osmolality. Osmolality changes can affect the gene expression and genomic imprinting also. This is how you should prepare a dish. You should make drops, cover with oil, again aspirate the drops back, and again load fresh media into the dishes. And on the temperature of the laminar as well as the airflow should always be switched off when you're making dishes. Always optimize the uh, macro and micro conditions to minimize the stress. A strict quality control is recommended with the help of gadgets which are validated with the National Institute of Standards, Mumbai. So if you are using a CO2 analyzer, it should be calibrated at an external agency. Then only it can calibrate your incubator, and you should do it at least once a year. Temperature, CO2, and oxygen should be measured daily. pH measurements can be done weekly or when you change the batch of media. Real-time loggers are preferred. Alarm systems are also mandatory nowadays. So there's a test known as sperm survival test, which can be run on, especially on instruments which are not mouse embryo tested. All the pipettes which you are using, serological pipettes, centrifuge tubes, culture tubes, and gases and filters, they are not mouse embryo tested. So when you are changing the source of these, you should always, or when there's a new batch coming in, you should always do a sperm survival test. I think we have to cover culture protocols also. So when you talk about culture protocols, I'll just take the gist of it. I'll not spend too much time on this. When you talk about humidified versus dry culture, then we'll talk about open versus droplet and single versus group embryo culture. We'll also talk about atmospheric versus low oxygen, which we already discussed that low oxygen is a better way of incubating. Cleavage versus blastocyst, and sequential versus single step. So we'll just take the talk about the take-home message in these. We talk about low oxygen. Low oxygen is preferred, and it takes care of high ammonium levels, and also it takes care of the increase ROS in the culture conditions. So this is a study which says that live birth rate, the ongoing pregnancy, implantation, blastulation, everything was found to be better in low oxygen compared to a normal oxygen incubator. When you talk about single step versus sequential, there has been a lot of studies, but recent Cochrane reviews have shown that we really can't choose one media over the other. Whichever suits best in your lab, use that media. Blastocyst and cleavage, there have been many advantages and disadvantages. All of you are aware of it. Blastocyst improves implantation rates, reduces multiple pregnancy. It's a physiological way of transferring the embryo. You can take a biopsy from day 5, day 6 embryo. There's a less risk of aneuploidy. There's less mosaicism. Disadvantages is that there's a high degree of cycle cancellation, monozygotic twinning, you lose on cryopreservation, and possibly there are epigenetic effects which are associated with prolonged cultures. So these are the things which you have to consider when you talk about humidified versus dry culture. Humidified is being preferred nowadays because the changes in osmolality and pH are minimal if it is a humidified culture compared to a dry culture, in which you have to maintain the right thick oil layer on the top of small media drops. So the first baby was born in an open culture. Edwards did it. But again, the side effects are the evaporation is much greater, the contamination to exposure to microorganisms, the penetration of VOCs, etc., is much more in an open culture compared to a drop culture. And it also dilutes the autocrine and paracrine factors. It is an embryo is like a small fish in Pacific Ocean. So if it has to talk to another fish, it is very difficult for it to talk. But if it is in a small drop, if it is in a bucket of you know two fishes are there, they can talk to each other. So similarly, in a drop they can, but in a well they cannot. So when you talk about droplet, and the chances of losing the embryo in drop is much less compared to the chance of losing an embryo in a well. And the thick oil layer, it takes care of the VOCs, it takes care of the microbial contamination. The small drop, there is a concentration of metabolites such as autocrine, paracrine factors, which stimulate the growth of the embryo. So the group culture also works better in droplet conditions. So VOCs also tend to partition in the oil phase and at much higher concentration in the oil, but also because the volume of the oil phase is so much greater than the volume of the aqueous phase that the VOCs are diluted. So the VOCs get diluted or are trapped in the oil phase and they are not able to reach the liquid phase. These are the special dishes which are available, and they've got if you make a normal drop and layer it with oil, there is a high degree of exposure of the media to the VOCs. But if the drops are small and there's a thick oil overlay, like in this, the drops are small and there's a thick oil overlay, the chances of VOCs penetrating this is much lesser compared to the other way of culturing. As the surface area of the exposed oil is smaller compared to the depth of the media and the oil. So these dishes are preferred, or else if you're using normal dishes, you have to cover with very thick layer of oil. When you talk about single embryo culture versus group embryo culture, there is no consensus which is better and which is not better. Each has its own advantages and each has its own disadvantages. We generally prefer group culture, in which we culture around up to 3 embryos in 40 microliters of media. But then again, there is no consensus on the optimal density of embryos in a culture drop. Between box and benchtop incubator, benchtop incubators are preferred because of faster recovery, and also because of the reason that in benchtop incubators, or the 100% gas is supplied from the cylinders, and in the cylinder you can attach an inline filter and take care of VOCs. Whereas in box incubators, only 6% CO2 is supplied by the cylinder through a inline filter, whereas 94% gas is being taken by the ambient air. The ambient air is very difficult to control, and you may not be having the right tight control on the ambient air. So it is always better to have a benchtop incubator for prolonged culture. When you're running a lab, you should always be very much focused on how to manage the incubators. There should be separate incubators for incubating the media, for transfers, and working incubators should be different from incubators which are used for long embryonic culture. So, in summary, I would like to conclude that attention to detail and adherence to protocols and quality control are very essential for optimal results. IVF culture media is an integral part of embryonic culture system, and it must be optimized in every lab. pH, osmolality, and temperature should be carefully monitored and maintained. The choice of oil is critical, which oil to choose and, how to use it, how to keep it. The logistics are also very important. And consumables and equipment of the highest quality should be used and tested regularly for its optimal performance in the lab. With this, I end my presentation, and I would also like to introduce that this coming 24th, we are starting a course, Embryology Demystified. It's especially meant for a better understanding of embryology for clinicians as well as for the embryologist. And in this course, we will be talking about how to set up the lab. I mean, what are the building blocks of a lab? In terms of the legal part, we'll cover the infrastructure, equipment, and stuff. We'll talk about the air quality and the clean rooms, and we'll also talk about the right consumables, media, embryology for embryology, and also we'll talk about the clinical factors which are influencing embryo quality. We'll talk about embryo grading, oocyte grading, sperm grading, and there'll be a huge discussion on clinical embryologist partnership and how we can nurture the relationship and how we can have excellent results in a lab where the relationship is also excellent. Each session will be followed by extensive question answer sessions. And we'll also talk about the quality control, the KPIs, key performance indicators, how to maintain an IVF lab, how to select the right equipment. And we're also getting faculties from the industry, the best people in the industry who understand the equipment, who understand the air quality. They are our guest faculty for this course. And in the end, we will finish with we'll talk about the post-thaw embryos, how long to culture them, how frequently to assess the embryos, day 1, day 3, day 5, day 2, or each day you have to assess them. In the end, Dr. Lakshmi will be touching upon the IVF lab errors, which are the common mistakes, and how we can avoid them. And each day will be followed by a interactive question and answer session, and in the end we'll have some MCQs as a feedback to the faculty members. This course is very nicely designed, and it is has been done last year also by the Fertilis Academy. It was very well attended last year, and I'm sure it will be attended quite well this year also. So I encourage all of you to join this course. It's a very, very useful and important course for a better understanding of embryology concepts. Thank you so much, and I'm open to questions now.
Thank you so much, sir, for the excellent presentation. You have covered everything from top to bottom about embryo culture and culture system. So I don't think anything is left over. So let us move on to the question and answer session. Sir, we have one question, sir.
Yeah, please.
Possible to sperm after preparation in MOPS medium because we haven't CO2 incubator. Please, what is the ideal temperature for sperm before
Yeah.
and after preparation?
So generally, we don't keep the prepared semen sample if we are preparing it in MOPS and HEPIS media. We don't keep it in CO2 incubator at all.
So
It is
they are not having CO2. So
yes,
can I
it is
use
it
MOPS?
yeah. They are not having CO2 incubator, no? So it is good that they prepare it in MOPS and HEPIS media, and they keep it outside at room temperature, or they keep it at 37 degree for 5 minutes, and then you bring it back to room temperature. So at room temperature, the pH is maintained for a longer time. The DNA integrity is also maintained for a longer time compared to 37 degrees. So you don't need a CO2 incubator in case you are only, it seems they are running an IUA clinic and they're preparing samples and they don't know where to place the sample. So the ideal temperature for sperm before and after preparation, best is room temperature only. So you don't need an incubator if it's a purely IUI lab. Lakshmi, your views?
Sir, at least to promote liquefaction, I feel
Yeah.
incubator
So that's different. That can be done even on a stage,
preparation alone. As
on
you said,
another stage also.
37-degree will activate the
Yeah,
sperm
yeah,
spinner.
much faster.
Integrity will die faster. So when you are planning to inseminate as early as possible, 37 is okay. Suppose if you're going to delay insemination or ICSI, whatever it may be, keep it at room temperature.
Yeah, and in your case, since you're preparing and it seems you have a IUI clinic and not a IVF clinic, you can always, prepare the sample and mix it and give it to the clinician.
Yes, sir. And many of them for liquefaction also, sir, people suggest that to see the liquefaction at room temperature. I feel it is not so. What's your opinion on that?
WHO says that for the initial 5 to 10 minutes, you have to keep it at 37 to promote faster and the timely liquefaction. And then after half an hour, you assess for liquefaction.
Yes, sir. And what is the best gradient for sperm wash in IUI? They've asked about this.
Yes, gradient for sperm wash.
I think they're asking about single or double, and what is the percentage?
So the percentage, it depends actually. We have used 90 and 45, we have used 150, and we have
80.
used 80 and 40. So I don't think there is a much of a difference. There are not many articles on which is the best gradient. So you can use any of these. Anything which is easily available at your center, you can use it.
Sir.
Lakshmi, your opinion? Have you tried and doing any research on this?
Nothing, I couldn't conclude anything. Everything is okay, sir.
So we used to buy gradient and 100% gradient, and we used to dilute it with media, make it 150, and that worked well for us. And now we are using 80 and 40, and previously we were using 90 and 45. So I don't see much of a difference between these.
Okay, sir.
So any more questions? Anybody would like to switch on the mic and ask directly? You're more than welcome. Can you switch on the camera for a group photo or something? Can we have a group picture? Dr. Supriya has got a question. Dr. Supriya, you have a question? You have raised your hand.
Yes, sir.
Yeah, please ask, ma'am.
Sir, good evening.
Good evening, Dr. Supriya.
Sir, I just want to ask one thing. They say after if you keep the semen sample, suppose for IUI, then semen sample should be kept at room temperature or 37-degree on that incubator, that
See,
warmer.
yeah, so it is good if you how we do it, I can tell you. So I cap it tightly. There's some noise. We Yeah, can you hear me?
Oh, yes, sir.
So we cap it tightly when we prepare the sample, and then we keep it in the incubator for at least 5 to 10 minutes. And suddenly, when it's exposed to 37-degree, there we see that the sperm motility increases. And especially when you're inseminating it for IVF and you want to assess the right degree of motility before inseminating, it is always good to keep it at 37-degree for at least 10 minutes. So that even when you're examining for motility, it is said that the stage temperature should also be 37 degrees. So it is a good idea to, and since we are doing quite a lot of IVF also besides ICSI, it's always a good idea to assess the motility the way it should be assessed. Rajiv Ranjan, can you just mute yourself? Okay. The recording will be posted on our YouTube channel as well as it will be posted on our app by tomorrow afternoon, maybe around 12 o'clock Indian time or maybe 1 o'clock Indian time. It will be posted.
Yes. Hello.
Yeah, please.
Yeah. Sir.
Yeah, please.
No.
Yeah,
Yes,
please.
sir.
There's a question?
Yeah. No, just I am belongs to my state, my Bihar. That is the city is that city is Motihari. And here IVF facilities it is already going to started, but thing is that how to develop for the best of like where in the Punjab and Delhi position is there. So this type of facility I have need to by your suggestion, sir. And that doctor same Dr. Sina and doctors. So I have invited to you to please come to one time in this place wherein here a lot of patients here.
Okay, Rajiv. If you have my number, you can call me directly. We can discuss about how to establish your center. And we do offer services of
Yes, sir.
installation and troubleshooting and managing a center. So we can discuss on the
Yes.
phone and we can take it forward. है ना? आप एक बार मुझे, if you can share your number or if you have my number, you can message me directly.
It is my group, sir, but I have a good number, so I think
I am putting in the chat.
man, then I can do it for proceeding.
Yeah, your voice is also not very clear.
Because here a lot of patient is, yes, because here a lot of patient is here, because this location is India Nepal border, and that is a very famous uh, sir.
Rajiv, you can share your number. We can write down your number and we can get in touch with you. Please share your number.
Sure, sir. Yeah, 889
Sorry.
8789
Huh?
2190
Uh-huh.
98
9?
8.
Okay. Rajiv,
Yeah, I'm repeating, sir.
no, it's fine.
8789.
It's fine. We have your number.
I'm just
You
repeating.
can message
Thank
me
you.
directly, or somebody from our team will call you also. Okay?
Sure, sure, sir.
We'll help you out. Don't worry. Any other questions? Anybody has any concern?
Yeah.
Any quality issues or any troubleshooting issue? Anything with the culture system? You can ask directly now. And the course which we have introduced is a very useful course for all of you. And it's a very reasonably priced course. I think it's 5,000 rupees only. So I encourage you to join the course. It's especially meant for clinicians and embryologists. Dr. Sunday, do you have any
Sure.
questions? Lucky, are you there today? Yeah, Lucky is there. See, he is sporting his Fertilis Academy t-shirt also. So how are you doing, Lucky?
Sir,
How are you doing?
how are you doing? I'm doing fine, sir.
So we can't see you on the screen. Something wrong with your camera?
Okay.
We want to take a picture of you with this t-shirt. Excellent.
Okay.
So when are you coming to India to attend our hands-on trainings?
Thank you, sir.
I said, when are you coming to India to attend our training?
By next year, sir.
Okay. Okay. You're welcome.
Yes, sir.
Okay, Supriya, how are you?
Sir, fine.
Center.
So wish you happy Diwali in advance.
Yeah, happy Diwali to all of you. Yeah. We might have another session very soon, maybe after Diwali or maybe before Diwali. We'll decide. So please attend the session also. If there are no more questions, we can end the session now. And if you can have a nice evening pre-Diwali and a good night to all of you. I'm ending the session. Thank you so much. Dr. Lakshmi, any comments from you?
Nothing, sir.
Okay.
For
Thank you.
cover everything, and I also request everybody to join the course and get benefited.
Yeah.
Thank
And
you,
the
sir.
next session is also going to be very nice and we'll cover the media components in details, great details. I've just touched upon superficially, but Dr. Lakshmi will be covering everything in detail. Thank you. Thank you. Good night.